Good afternoon. I'm Chris Tompkins, CTO of KromaTiD, and with me today is Erin Cross, our Director of Whole-Genome Development. We'd like to welcome you today to the second of our seminar series focused on the importance and value of genomic structural measurements. Our first seminar was focused on localized measurements of genomic structure and structural variation using directional genomic hybridization, or DGH. These localized tools are ideal for mapping edit sites, mapping genes, tracking lentiviral inserts, and are branded as DGH Insight. Today, however, we are delighted to bring to you DGH SCREEN, a direct, whole genome, unbiased, single-cell measurement of genomic structure and structural variation. As you will see when Erin dives into the studies, DGH SCREEN is an efficient tool for detecting DNA damage, discovering structural variations, and for QCing cell lines, clones, and the therapeutic products of cellular engineering. Before we dive too deep into technology and data, I thought I would take a moment to introduce the company, KromaTiD. Based in Colorado, KromaTiD is dedicated to genomic structural analysis, measuring structural variation and understanding the role genomic structure plays in human health and healthcare. We today work with over 20 pharma and biopharma companies, plus institutes like NASA and the NIH, providing data that helps them measure DNA damage, understand off-target effects of their CRISPR edits, and track the distribution of their viral inserts. And we hope speed novel gene editing-based therapies from R&D to the clinic. We are able to impact therapeutic gene editing and lentiviral programs because DGH provides unique data that is both actionable and orthogonal to NGS and the other typical research and QC methods. DGH is a metaphase hybridization mapping method using synthetic DNA, single-stranded fluorescent probes to paint chromosomes as illustrated in this cartoon. The trick is that for DGH, we render each chromatid in the metaphase single-stranded by stripping the daughter strands, leaving 2, not 4, strands of oppositely oriented DNA in the modified metaphase. We then design our hybridization probes to give a signal pattern that matches the reference genome. Any change in the signal from this normal pattern indicates a structural variation. When we design the probes for a small region, say an edit site, this is what we call DGH Insight. When we design the probes to cover the entire genome, or at least individual chromosomes within a genome, that's our DGH SCREEN, the subject of today's seminar. When we work with gene editing companies, we often get the question, why can't we use a sequencing method to measure all of the unwanted byproducts in our system? The simple answer is that byproducts arise from 2 mechanisms, misrepair and misediting, that lead to distinct analytical challenges. The more familiar mechanism, misedits, typically generates small indels at the edit and off-target sites. These small indels can be effectively predicted and sequenced using a targeted amplicon method such as GuideSeq. Misrepairs, however, occur when the cell's naturally occurring DNA repair mechanisms, homologous and non-homologous end joining, reconnect the wrong ends from 2 different double-strand breaks, leading to not an indel, but a structural variation, a rearrangement of kilobase or megabase sections of sequence. Because these structural variations are large, very heterogeneous, and low prevalence, they are extraordinarily difficult to either predict or to sequence. As Erin will show in just a moment, with unbiased, single-cell whole genome DGH SCREEN, measurement of complex structural variations is not only possible, but routine and reliable. Before I pass the mic to Erin, I did just want to briefly show DGH in action and also to point out one additional feature of DGH SCREEN. Shown here in the center is the genomic map of a single cell. In addition to a direct measurement of structural variation, DGH SCREEN is also an excellent karyotyping method. So with a single assay, you can detect not only all of the structural variation within a system, but also karyotypic defects such as aneuploidy or trisomy.
Thanks, Chris. And thank you all for joining us. I've been working on DGH SCREEN for over 10 years, and I'm truly excited to show you the power of KromaTiD's first whole genome DGH assay system. With that, what I'm going to focus on is the nuts and bolts of how the assay works and a few demonstrations of the power of the assay. Illustrated here is a cartoon version of DGH SCREEN. As Chris mentioned, we prepare our unique metaphases by stripping the daughter strands, leaving chromatids with single strands of oppositely oriented DNA. Our hybridization probes are single-stranded, fluorescently labeled, and are designed to target a single chromatid in these metaphase chromosomes. For DGH SCREEN, we've designed millions of probes to target the unique sequence across the entire human reference genome. What our cartoon illustrates with these unbroken signals on a single chromatid is the genome of a single normal cell without any structural variation. Chromosomes can be identified and counted by color, shape, and centromere position. Measuring structural variation in this system is as simple as tracking changes to the signal pattern. Signals changing from one chromosome to another are translocations and insertions. Signals moving from the painted side of the metaphase chromosome to the unpainted side of the metaphase chromosome are inversions. Typically, we talk about DGH SCREEN as a single-cell assay, but more accurately, it's a measurement run in hundreds of single cells from a sample to identify constitutive structural variations and to profile heterogeneity, however complex the genome might be. What types of analysis can be run using SCREEN, and why is SCREEN data useful or important? On the most basic level, SCREEN is a general assessment of genome integrity and stability. Using classical cytogenetic assessments, along with the DGH advantages of strand-specific event detection that Chris explained earlier, the complete genetic picture of a sample can be constructed. The per-cell variant profile, which includes both random events as well as clonal events, can be analyzed for the population-level reporting of germline events and any subclonal outgrowth or mosaicism present in the genome, alongside the overall rate of background misrepair happening in the cell. Highly unstable samples can be flagged, and samples that are karyotypically changing over time due to selective growth advantages or transformation can be identified. The whole genome can be screened for previously undetected constitutional variants that could be present in the background of an editing system or even present in a rare disease patient as the subject of study. SCREEN can be used to investigate confounding sequencing results or low confidence calls, which often present themselves when analyzing structural variants greater than 500 BP with NGS. Now let's talk about what this assay is in practice. Here's how I sort and identify the chromosomes from a single cell. Metaphase spreads prepared for DGH SCREEN are imaged and then arranged into karyograms based on the color and shape of the painted chromosomes. I then identify the variants present in the cell based on the changes in expected signal patterns. For instance, on chromosome 1, there are 2 small inversions and 1 homolog of chromosome 4 is missing. To elaborate on the value and utility of DGH SCREEN, I wanted to talk to you about some results from 3 studies we used to beta test this assay. The first experiment is a collaboration with the University of Connecticut to measure radiation-induced structural rearrangements in 3 Mile Island survivors. The second experiment involves analyzing clonal cell lines generated from an immortalized human fibroblast line exposed to several different types of radiation in collaboration with the University of Texas Medical Branch. And the third experiment is an analysis of the GM24385 cell line, the genome in the bottle line, widely referred to as the most sequenced genome in the world. That is a collaboration with the NIST Genome Editing Consortium. DGH SCREEN can be used to profile DNA damage from any cause. It's not just a technique to monitor the byproducts of gene editing. In a study overseen by the University of Connecticut, DGH SCREEN is being run on samples from a small cohort of individuals potentially exposed to ionizing radiation at the time of the accident. KromaTiD is also supplying DGH SCREEN to NASA to profile radiation exposure in the US Astronaut Corps. From data gathered in previous studies, it is known that structural variations present in the genome as a result of radiation exposure, especially inversions, persist and are measurable in the blood of affected individuals. The traditional method of biodosimetry is to measure structural variants on a small subset of the genome and extrapolate to the rest of the genome. This data comes from the respective biodosimetry study performed on exposed US atomic veterans and shows the dose calibration curve generated in control samples. An early version of DGH SCREEN used DGH chromosome paints 1, 2, and 3 in a single color biodosimetry assay, as shown in the image on the left. Event rates for both translocations and inversions were extrapolated to whole genome equivalency to account for the unpainted portion of the genome. Both inversions and translocations increased in a directly proportional rate to the dose of radiation, or, put another way, dose of double-strand breaks. Inversion rates increased at a steeper rate than translocation rates and proved to be a more sensitive unit for measuring received radiation dose. The ability of DGH screen to add inversion rates, even in a limited study like this, dramatically increases the sensitivity of traditional biodosimetry techniques. While this was a successful technique, imagine what could be done with a whole genome assay. We move now to some of the data generated from the 3 Mile Island study samples. The image on the left shows the metaphase spread before sorting in karyogram organization. And the image on the right is organized to reflect the chromosome ID per homolog into a karyogram for assessment. Chromosomes with structural variant events are shown circled in white. A reciprocal translocation between chromosomes 7 and 14 was observed in this cell, along with several inversions and sister chromatid exchanges. The 2 translocation derivative chromosomes have a combination of pink and green paint. This image illustrated the power and sensitivity that can be achieved by removing the extrapolation and directly measuring every variant across the genome. KromaTiD is currently collaborating with the University of Texas Medical Branch in Galveston to provide structural variant data for clonal derivatives of individual human cells surviving ionizing radiation exposure. For this second use case, I'd like to show you the DGH screen results from one of the clones. The karyograms here are from 2 separate cells in a clone derived from a sample irradiated with an iron source. In this clone, there's a recurrent non-reciprocal translocation between chromosomes 6 and chromosome 3, visualized as a combination of yellow and orange paint on the second chromosome 3 homolog in each image. The inversions in the normal chromosome 3 homolog and 1 homolog of chromosome 12 are new findings for this clone and are pictured in orange circles. The rest of the rearrangements, as you can see, vary between both of the cells and represent the background rate of misrepair happening in the cell line. In order to characterize the recurrent or clonal events present in a sample, several individual genomes must be screened and analyzed. This slide is an image of a cell from another clone we've run DGH screen on for this study. Without spending too much time diving into karyograms, there are inversions circled in white, a complex translocation outlined in an orange square, and a sister chromatid break outlined in a green triangle present in this cell. Next steps will be to identify all recurrent events in each clone for comparison to EMFISH, GBAND, and sequencing data. The first 2 cases, which I've just briefly touched on, are simple illustrations of the power of DGH screen. However, there is no limit to the level of structural heterogeneity, complexity, differentiation, or transformation that we can measure with DGH screen. The samples I'm going to discuss with you today were provided to us by NIST and were generated from the progenitor cell line of the genome in the bottle sequencing standard. These cells were donated by a rare and undiagnosed disease patient and were immortalized by Epstein-Barr virus transformation. The data I'm going to show you today illustrates the difficulty of working with these types of immortalized cell lines, which can become unstable in culture and differentiate or transform. The first set of DGH screen data generated on this cell line at passage 12 had a wide array of both recurrent and random variation present, along with some complex events, hinting at genomic instability. Circled in white is an additional observation seen in just over 60% of the cells, a prominent size difference between homologs, indicating a potential defect in chromosome condensation, also associated with genomic instability. Circled on chromosome 8 is a constitutive inversion observed in almost every cell. It's one of 34 inversions seen above 8% prevalence in this cell line at passage 12. Aneuploidy was observed in just over 20% of the cells. By passage 18, we are seeing a much different picture. Some of the same structural events were present as a theme, but there was a drastic increase in the rates of complex structural variants and aneuploidy present in the cell line. Pictured here and circled in white is a triradial chromosome 16 with a partially truncated chromosome 16 fragment. We also saw centromere spindling across several chromosomes, including chromosome 16. Centromere spindling is also tied to defects in chromosome condensation. And the size difference between homologs we observed in passage 12 at just over 60% of the cells was observed in over 90% of the cells by passage 18. Aneuploidy, mainly chromosome loss, had increased prevalence from 20% of the cells at P12 to just shy of 50% of the cells in P18. In the karyogram pictured on this slide, homologs of chromosomes 10, 14, and 19 are all missing. Chromosome 16 in particular was highly unstable, and events of every type were present. Examples of the recurrent inversions, translocations, whole arm deletion, triradial formation, and the centromere spindling referred to earlier are all depicted on this slide. Interestingly, the number and prevalence of inversions changed dramatically between the two passages. Of the 34 total recurrent inversions observed in passage 12, only 7 of those were still detected by passage 18. The germline inversion on chromosome 8, observed in 96% of the cells at passage 12, was present in only half of the cells by passage 18. These changes in inversion profile also indicate genomic instability, transformation, and the start of subclonal expansion among the surviving cells. To confirm some of the more striking karyotypic DGH screen results seen in this passage of the cells, we performed G-banding on them. The instability and gross rearrangement of chromosome 16 was observed, as well as some rate of potential condensation defects. Translocation and aneuploidy rates matched the DGH screen data, and none of the recurrent inversions were detected, as they would largely be invisible to this method. So I know that's a pretty deep and complex data set, and I appreciate everyone hanging on with me as we dove into the details. What I want to do now is wrap it up and talk about both the implications of that data and the value of DGH screen for controlling and monitoring cellular engineering products. Despite the data complexity, what we were looking at is a relatively simple story, illustrated in this diagram. This particular immortalized cell line, generated from a rare disease patient, proved to be unstable and transformed dramatically over a few relatively early passages. The implications of this are that even if separate labs begin with the same passage, and thus presumably the same genomic structure, if they grow the cell line at all before analysis, they cannot assume that the structure from lab 1, lineage 1, will match lab 2, lineage 2. The other important thing to note is that this particular phenomena was only discovered by a direct, unbiased, whole genome single cell technique, DGH screen. Thank you very much.
Thank you, Erin, and I want to thank all of you for attending today. As I mentioned, we have links to our previous seminar in this series concerning DGH Insight on our website. And please be on the lookout for our next seminar for a new ChromaTiD product, DGH Discover. DGH Screen is available today, and we will be delighted to partner with any and all of you to help solve your genomic analysis problems.